All right. We've been talking about how to make a body plan, and we're almost through that. But it's worth recapping. So, mom makes an egg that is non-homogeneous, and once it gets fertilized, the cells start dividing. In the case of Drosophila, the nuclei are dividing, and there's no cells yet. But eventually, they're spread out along the body, and we talked about a rostral-caudal signal to tell each cell approximately where you are in the rostral-caudal axis of the individual. And we were just getting to, and therefore what structures they should make. And we were just getting to the dorsal-ventral. But within that, in terms of Drosophila, we talked about the cascade of genes, starting with maternal polarity genes, right? That are going to give rise to many good gap genes, people, pair rule genes, expect or seek for the semi-ganglion cells. That's not it. The genes. Segment gene, segment polarity gene. Thank you very much. You should teach this class. And that cascade means that eventually virtually every nucleus, or take any tiny chunk of nuclei there together, and they will have a history of transcription factors that they have been expressed and that have been exposed to that's unique from any other tiny cluster in the syncytium, and that's going to tell them what they can make. And it's only that last tier, the Hox genes, where mutations there can provide a last-minute switch in direction, where instead of making an antenna, because they didn't get the Hox gene, they made a leg, right? And so that's how you can end up making a cute little fruit fly to pester you. And they've been doing it a long time. I'm not pestering you, but making fruit flies, right? Each one of which works just well enough to make more fruit flies. Okay so far? So we were talking about the when we stopped, we were talking about the dorsal-ventral gradient signals. And I want to get back to those, where the notochord is releasing sonic hedgehog, which is telling the bottom of the neural tube, the ventral part of the neural tube, to make a floor plate. Overhead is our skin cells. They're continuing to make bone morphogenetic protein, and they're telling the top of the neuraxis of the neural tube to make the roof plate. And that's all going to have consequences for what kind of cells are going to develop there, because it's going to be down here in the floor plate that some cells are going to make motor neurons. They're orange here and here. Some cells are going to make motor neurons in the ventral portion because of sonic hedgehog signaling first from the notochord and then from the floor plate itself. And other cells in the top are going to start making sensory cells, sensory assemblies to take in sensory information. And there's several different, again, there's redundancy in the dorsal signals. And here's some of the names. And eventually there ends up being a gradient of TGF-beta bone morphogenetic protein-like signaling from the dorsal to sonic hedgehog from the ventral. And again, within that dorsal ventral position, within, let's say, the spinal cord, different neurons are going to arise because they've gotten different instructions about which genes to express and therefore what structure and function to take on. Oh. One more topic that I think is interesting. Sonic hedgehog, remember, it's going to tell the bottom of the neuraxis to make a floor plate. But in the front of the brain, remember the eye cups that Bergmann studied? The eye cups that come out are going to hit the epithelium and induce it to make lenses and stuff, corneas and the like. Why are there two of them? Well, it turns out that the notochord in the front, on the front part of the brain, they respond differently to sonic hedgehog. They avoid it. And so a midline appears right there where the notochord is. And so instead of one eye cup coming out, two eye cups come out. Everyone get that? And that's why you could have two eyes. It sometimes happens that pathway for sonic hedgehog to inhibit growth, it sometimes happens that a gene is present where that's not there. That signaling doesn't work. Doesn't matter. There's several different ways it can happen. It doesn't matter. The important thing is the eye cup isn't getting this signal to be inhibited and split in two. And in that case, only one eye cup forms. And that then has a cascading effect because it turns out that same signal isn't just telling the brain to make two eye cups. It's also important in organizing the face and the mouth and things like that. So when this happens, it is disastrous. It is not survivable, right? Often the fetus will make it all the way to birth, and now it has a problem because usually there's no mouth system to take in nutrients, for example. And the result is that split in the eye cups, that inhibition that causes two eye cups to come out, is also going to be responsible for inhibiting growth of the cerebral cortex. And so you end up with an individual, instead of having two cerebral hemispheres, has only one great big cortex. Again, not survivable. And so when this happens, and it happens in domestic animals, occasionally it happens in people. This is such a brain from a person. Then we end up with, do I have the term here? I hope I do, holoprosencephaly. So having only one big cerebral hemisphere is known as holoprosencephaly. And it's fatal. Questions about that? And as I say in the book, I'm sure all those legends about Polyphemus, right? The Cyclops that pesters Odysseus and his men, eats them and all that stuff. I'm sure ideas about such monsters are because people were aware occasionally a child was born this way and died, either a child or livestock animals. So that's how people knew, oh, sometimes individuals have only one eye. What if there were a whole race of people? Okay. So remember I told you that in the next edition of the book that you're just a little too early to get. I end each chapter with some more cutting edge sort of stuff instead of the basic things. And I don't want you to miss out on that. One of the new things that's going on that's really kind of taken over developmental neurobiology is the use of organoids. Actually, it's in all biology, where people have found that they can get cells, typically, for example, fibroblasts from muscle cells, any ordinary cell, and get it to grow in a dish, one that grows well in a dish, like fibroblasts. And then if you treat it with the right factors, you can get it to sort of start acting like an embryonic stem cell, or a multipotent, or at least pluripotent, maybe even totipotent cell. And once you have these cells growing that way, then if you're studying liver, you can expose them to factors that make them start acting like liver cells. So you've got this isolated liver in a dish. Or there are other ways you can treat them where they start expressing neural genes. And so you have these neurons, disembodied neurons, kicking around there in a dish. And when you're doing for neurons, they're known as organoids. The clumps of cells are known as organoids, organ-like, and these are brain organoids. And the reason there's so much excitement about it, first of all, they're dead easy to make and they're cheap, right? Because you don't have to have any animals around. But another reason why they're exciting is it's like you finally have a lot of control about what's happening. And it's pretty interesting how many things these isolated neurons in a dish or neural precursors in a dish will do that reminds you of basic neural development, including making a hollow sphere, right? That would eventually make a neural tube if it had some more structure to it. And then they start expressing neural genes, and they start differentiating, where some take on a neuronal fate and some start taking on a glial fate. Have I said this yet? If I'm saying it right, neural, Neural, that refers to either neurons or glia, any old brain cells. But when I say neuronal, I'm referring only to neurons, okay? So that's the more specific. And when I only want to say glial, I'll say glial, right? Anyway, so one of the things that people have been very fascinated with, we've been, sequencing the genome of Neanderthals for quite some time now. When I was in graduate school, I was still a professor at Berkeley. The story was that Neanderthals, clearly they had existed. Interestingly, they had larger brains than Homo sapiens, but we know they disappeared. And in those days, the idea was the Homo sapiens outcompeted them. And maybe what that means is, people did what people do. They killed them all, right? That's, plenty of examples of that in prehistory where that's what a group of people do when they get to a new place and there's anybody not them that's there. That's just what we do. But it turns out that in fact, there was sufficient interbreeding between them that many Neanderthal genes are in fact in circulation today. So they weren't totally separate from each other. And you can even pay services that will tell you, I'll let you know how advanced I am. I have like 70% of my genes supposedly have a Neanderthal origin. Yeah, I know, which explains the hard head. And anyway, but people have been cataloging, and there's some genes that are quite different. And so this group, they zeroed in on a gene called NOVA, and they noticed that there's an allele in Neanderthal populations that they don't find in human populations. And so they suggested maybe it was the mutation of this allele that caused or led to better brain development in Homo sapiens than in Neanderthals. And it's not bigger brains, right? Because remember, the Neanderthals had bigger brains than we do, and they're all gone, but that it might have made them better brains. And we really don't know anything about the structure of the Neanderthal brains because brains don't leave fossils, right? There's nothing there to study. And so they said, well, what happens when we make brain organoids and we either give them with the human version of NOVA1 or we give them the Neanderthal version of NOVA1? And can we see a difference? And indeed they did, which is shown here. So this is a complicated graph. Did I get I didn't get the new version in time to show you, where they're keeping track of two different synaptic proteins. One is known as SYN1, synapsin1, and the other is postsynaptic density 95. We'll talk about those when we talk about synaptogenesis in a couple of weeks. So at this point, they're just markers for how much synapsing is going on between these various cells in the organoid. And here, if both copies of the NOVA1 are human, you get this amount of synapsin and you get this amount of PSD 95. And I'm going to skip over what happens when you look at knockout. But when you have it be homozygous for the archaic, which is to say Neanderthal version, in fact, there's less synapsin going on and less postsynaptic density 95 going on. They proposed then that what's going on is this change in NOVA1 meant that we didn't make bigger brains, but maybe we made more synapses between the brain cells we've got, and maybe that made all the difference, which is why we're here and they're not. How's that? Worth studying. I mean, on the slides. And I think that I put some text in the, did I? Yeah, I put some of the text I wrote up to describe this on the D2L site, right? So you'll find it there under the class notes. Questions about that? We'll run into brain organoids again. They're cool. So to summarize from chapter 2, unlike C. elegans, vertebrates, and almost all the other animals on this planet, development is one where the cells are constantly talking to each other to decide where they are in the body and therefore what particular gene expression they should take on, which will mean what sort of structures they'll make. And when everything goes the way it's usually does, then you end up with a whole organism, right? With all the parts in the right places, including a brain with all the parts in all the right places. This communication takes, is various kinds. Sometimes, as in the syncytium, it's one cell making transcription factors to affect its neighboring nucleus, for example. Or it could be that cells are there and one cell expresses something on its membrane that affects the gene expression of its neighbor, and they're all talking to each other. Some factors are diffused, like the bone morphogenetic protein down from the epithelium, or sonic hedgehog going up from the notochord. But what they all have in common is the cells are constantly keeping track of each other and directing each other. And as with the delta notch system that we saw in flies, it can also be to the extent where they'll be touching each other, and one will decide to become a neuron and the other becomes skin cell. Or when we get to the mammalian spinal cord, that same system will mean some cells are going to become a neuron and others are going to take a lesser role, if you like, think of it that way, of being a glial cell. And suddenly you have the most complicated machine in the universe sitting between your ears. Maria?
About brain organoids.
About brain organoids, yeah.
So, different factors can be given to help these stem cells end up getting to differentiate into a certain fate of becoming a neuron. What different factors have to be given? For example, if you want a neuron that's an inhibitory neuron, or different parts of the brain.
So, as far as I know at this point, what they know is which factors to mess around with to get them to go down the neural pathway. But of course, mostly that's been figured out through trial and error. What chemicals do you have on your shelf? And you dissolve them and you see what they do, right? And you try to keep track of as many different growth factors as you know about. Well, there's tons of those. Epidermal growth factor, there's a whole bunch of them. TGF beta, there's a bunch of them. And so for the most part, it really is a matter of, well, let's try this. And so it's all been by trial and error. And in fact, no one has any pretension that this is the particular pathway that neurons normally go through. It's just a pathway that works, and now you can start studying the cells. Very, imperfect system. On the other hand, if they keep at it, maybe eventually they would, be able to figure all that out and know what happens in vivo, not just what happens in a dish. Okay. There was something else I wanted to tell you about this. To go back to holoprosencephaly, imagine you have a baby and it's born, and it's right away, everyone knows this baby is not viable, and it's kind of monstrous to look at, and you may or may not want to look at it, right? Imagine then you sit down with the parents afterwards and they want to know what went wrong, and you say, oh, we know what went wrong. There was a problem with sonic hedgehog gene. Not, why is it called sonic hedgehog? Oh, well, it's a joke.
Fun.
Not so funny when it's affecting your kid, right? And so there's a movement afoot for genetic counseling and things like that to start getting rid of those whimsical, funny names that were so much fun for biologists to work with, and occasionally helped us remember what a gene does, and that maybe it's time to rethink the nomenclature of these things, right? Because, it was different when they named them in flies because nobody knew anything about these things in humans, right? Nobody knew that they had consequences. But the more we learn, the more insensitive it seems to be doing that. Okay, let's have a Kahoot then. Don't be jealous. Just, compete more. Okay. So, in chapter 3, we're going to talk about vertebrates primarily in this chapter, and where have we gotten so far? Well, we've got the neural tube is formed, right? And because of one set of inducing factors dorsally and another ventrally, different cells are getting different bits of information, and they've been specified in terms of anterior, posterior position. And at first, the neural tube is just one cell thick. But of course, you can't make much of a nervous system that way. And so what happens next is the cells there start dividing, and what we're going to be talking about in this chapter is neurogenesis, where lots and lots of neurons get generated. And then they get generated in one place, and they have to migrate to get to the right place. So this topic is neurogenesis and migration. So preparatory to that, a little bit about pleiotropy and redundancy, which we've talked about briefly, but I just want to emphasize it again. So pleiotropy is when one gene, like Sonic Hedgehog, has impact on lots of different traits, like the formation of the floor plate, the formation of motor neurons, the formation of eye cups, and whether or not there's going to be Holoprosencephaly, right? So lots of different, but then it also has consequences for whether a mouth gets formed, right? Now, if you actually understand all the mechanisms that are involved, it may be that there's one particular thing that gene does that is responsible for all those different traits. But for the most part, when we find out a gene, a mutation of a gene causes some problem, for the most part, we don't know the mechanism, and therefore we don't see why there's a connection. And so here, eventually, we're going to see how mutations, one set of mutations, several different genes involved, can end up with someone who can't hear very well and can't detect smells. Hearing and smelling, they sound like they got nothing to do with each other. Not only that, they don't undergo puberty when all their peers do. So there's three things that seem totally different from each other, and they're all being influenced by these several genes, right? Each one of them influences. So that's an example of pleiotropy. And for that particular example, the reason I'm using it, of course, is we're eventually going to understand the mechanism that will tell us how, there is something in common with puberty, smelling, and hearing. You okay? Okay. And we've seen several examples of redundancy, and I would say that redundancy of cell signaling is much more common in vertebrates than it is in invertebrates, right? So one of the things that's different about fruit flies and people is we've got 4 sets of the Hox genes, and the fruit flies only have 1, and that's probably because we're more complicated. Our bodies are more complicated than theirs are, and therefore one consequence of those more genes and more complications is occasionally there's redundancy. Okay. In the 19th century, as anatomists are making use of this cool thing called a microscope, nobody knows who invented the microscope because there are several different legends about it, but eventually people found out that you could see things that were really close, and we talked about that some when we talked about Robert Hooke and what he found, etc. But right away, they could look at the developing nervous system. And what they saw when they looked at the neural tube, they saw pictures like this, where there's the outer part of the neural tube, which is eventually going to give rise to the pia, and so we'll call that the pial surface. And there's the inner portion of the neural tube that has the hollow point in the middle. That's a lumen. That opening in the middle is just a lumen. And as I said, at first, this neural tube is only 1 cell thick, but eventually there come to be more cells, and right away, anatomists noticed something about what they would see here, which is that they noticed that on the, and this is also sometimes known as ventricular surface, because this is eventually going to be the interior of the ventricles. They noticed that there were these strange things they could see with their staining methods, where some cells were showing these spider-like figures that were as if they were pulling, they looked for all the world like 2 spiders, or a spider on a mirror, like a spider that was on a mirror and was pulling itself up, and getting stretched out. And of course, they can't see any of this in living cells. All they can see is what's dead stuff. But they realized that these were chromosomes and that cells were going through this. And so they inferred that probably these cells were dividing. And that in dividing, they were somehow duplicating chromosomes and divvying up the chromosomes between the two of them, right? And so they called them mitotic figures because they already called that mitosis. And this was one of the clues that maybe the chromosomes have got something to do with inheritance, but nobody knew yet, right? That was the leading theory. And then it would be, 100 years before anyone figured out what the chromosomes were made of and how they encoded for amino acids. So far, so good. And again, looking at looking at images at different stages, they eventually also intuited that what's happening is the mitotic figures only happen at the bottom, but that the nuclei are actually shuttling back and forth between the lumen and the peel surface in a manner like this, where they keep these cells. At first, they have connections both to the peel surface and the ventricular surface, and they don't let them go. And the nuclei are shuttling back and forth within the cytoplasm, sometimes near the top and sometimes at the bottom. And they only showed mitotic figures when they were at the bottom. And eventually, as development progresses, they find some cells that have neither connections to the top or the bottom, and they're just hanging out there. And they never saw any mitotic figures there. And so they figured those are post-mitotic cells. And they're going out and they're going to form the rest of the neural tube, and someday they're going to make neurons and glia and stuff like that. And so we call this sort of division, symmetrical division, where at first they're dividing, and now we've got two cells that are touching top and bottom. And they just keep dividing and doing that for a while. But at later stages of development, you see asymmetrical division, where the division takes place and one cell doesn't duplicate the connections to top and bottom. It's free floating and it starts moving around. And yeah, the cells have all the machinery to move around, and we'll talk about that quite a lot later at the end of this chapter. And all of this you've heard of before, right? Yeah. We call these cells that are busy dividing, we call them neuroblasts, right? So, because they're going to give rise to neural cells, neurons and glia. And another view of this is that as development proceeds, you end up eventually with this zone where there's no cells, just the processes, right? The nuclei might come up, but then they go back down. And it's only after asymmetrical divisions start happening that you see any cells that are marooned out there, no longer connected top and bottom surface, but now there's a clump of cells. And we call that, at this point, the marginal zone, because it's on the margin, and the ventricular zone, the part that's next to the ventricles, what's going to be the ventricles, where all the dividing is going on. And as development proceeds, what eventually happens is there's a marginal zone, and then there's an intermediate zone where cells are hanging out, and then that ventricular zone where cells are dividing and populating the marginal zone. Pardon me, the intermediate zone. Questions about that? Yeah.
On the deep window,
Pardon?
does it like whole zone?
No, the marginal zone is going to be through the entire cortex. What we're seeing here is every part of cortex there is, right? They all start out this way. And there will be, differences in development eventually, but they're all going to have a marginal zone as long as the animal's alive. Right? And yeah, good.
Does it work the same way when neurons are created like later past development, like in hippocampus?
You got to wait. Yeah, we will talk about hippocampus and adult neurogenesis. No, it's different there because there's a little population, isolated population of subventricular cells that are continuing to divide even though they're not next to the ventricle. See, this is why you can't ask now. Okay. So right away, well, I should say, once the worm people found that you could predict the neuronal fate of a cell based on mitotic lineage, the question arose whether this is also happening in vertebrates. And it's just that we can't keep track of the mitoses, and therefore we can't notice that always a motor neuron is the result of, this mitotic division followed by that, by that, by that. Right? So people devised ways to label cells near the end of mitosis and label their progeny to see what happens. And it was done in lots of different, in brain and spinal cord. And I was part of this, so that's why I like to talk about it. So where you could put into a developing chick spinal cord retroviruses that include a marker gene that happens to be called LAC-Z that makes an enzyme, beta-galactosidase. You don't need to remember either of those things, but you'll run into them a lot. There are a lot more marker genes, a lot more ways of labeling cells these days, but once upon a time, this is one of the few ways you could label living cells and have their offspring continue the label. So you put in some virus into the developing spinal cord. Most of the viruses never find a cell to infect, but a rare event will happen. Occasionally, a virus will find a cell and infect it, and it will be a cell that still has several more divisions to go through, right? You got to put it in the right time. Put it in too early, there'll be thousands of cells that are labeled, right? But if you go late enough in development, that cell may divide 3, 4, 5 times. And so there'll be a relatively small cluster of cells, all of which had as its great-grandmother that cell you infected with the virus. And because that cell you infected with the virus had the gene inserted in its genome, all of its offspring are going to express the LAC-Z. And there's a very simple reaction you can do. And so now you can see as the spinal cord grows up, find a little cluster of cells that are all showing this blue color. And therefore, they all derive from a single cell, a single progenitor. And wouldn't it be interesting if some cluster of blue cells, we can call them a clone, if some cluster of blue cells were all motor neurons, and another cluster of blue cells were all astrocytes, and another cluster were all oligodendrocytes? Would have been cool, and that's exactly not what was seen. So in fact, what was seen is that even though there were only a few more divisions going on, this particular clone, they all come from the same progenitor cell. Some made motor neurons, some made this kind of glia, oligos, some made astrocytes, some made neurons in the spinal cord. Or here's another clone where one made a preganglionic sympathetic cell, some motor neurons, some astrocytes, some oligos, etc. And so even at the late stage of development, what decides the particular fate of one cell versus another isn't a result of mitotic lineage. And by now, we know that what's directing them to take on these various fates instead is cell-cell interaction. And sometimes the cell-cell interactions are antagonistic, like in Notch delta, where the very fact that these cells just divided and they're right next to each other, that doesn't mean that they're going to have the same fate, because one cell might be expressing a little more delta and they might inhibit its neighbor so that this cell with more delta makes a neuron and that cell makes a glial cell. Yeah. And I guess I meant to point out that this question of whether there was a mitotic lineage, the problem was when you're working with these organisms, there's so many cells, nobody can keep track of them and they all look alike, right? So you had to take a different approach, which makes you think, gee, how cool that C. elegans had those asymmetrical divisions so that you really could catalog and keep track of all those, it didn't need to work out that neatly, but it did. So division's going on. The fate of these various cells is going to be depending on interactions they have with one another. And what sort of zones you see after the ventricular intermediate marginal zone depends a little bit on which part of the brain you're looking at. And because we're going to talk about both cerebral cortex and cerebellum, I want to talk about the differences between them. And this is sort of a preview where we're eventually going to explain how it is that these differences arise. But for now, I just want to get the terminology out. Okay? So here in the cerebellum, we still have the pial surface and the ventricular surface. And by the time we get this late in development, in fact, all neurogenesis is going to be happening at the ventricular zone. So that business of the mitotic figures being only next to the ventricle, that's going to continue. And in the cerebellum, there will eventually be the ventricular zone where neurogenesis still takes place. Right now, in your brain, in the ventricular zone, there are cells dividing like mad. Mostly they're making glia, but that's where all the division takes place. So they divide down there, and then they shinny out of the ventricular zone to go into the rest of the nervous system and do stuff. So in early stages of development, then, we have the situation where there are these glia that are specialized in the cerebellum that are known as Bergmann glia. And even when the cerebellum is quite thick, these little cells, being stretched out like a prisoner on a rack in a medieval torture dungeon, they still have one process attached to the pial surface and another to the ventricular surface. And they're not going to make neurons, they're glia, and they are crucial for the right development of the cerebellum. Because what's going to happen is as cells kicked out of mitosis down here from asymmetrical division, they start climbing up the Bergmann glia to get further and further away from the ventricular zone. And eventually, in the cerebellum, some of these cells go close to the pial surface and they develop into the largest neurons in your body, the Purkinje cells, which you might have heard of in the cerebellum. So Purkinje cells are huge. They're so huge, they're tetraploid. They've got two sets of chromosomes because they're so big, they've got to have plenty of access to transcripts and genes and stuff like that. So they're huge. I'll tell you a little bit about their morphology now. So at end, they are found only on the outer layer of the cerebellum, the cerebellar cortex. And you can think of a Purkinje cell as being shaped like a hand with the fingers splayed out, where palm of the hand is the cell body, and the arm beneath it is its axon. And those fingers that are splayed out, those are these huge dendrites that they have. And right away, when neuroanatomists started doing this stuff, again at the end of the 19th century, they were fascinated by the fact that these Purkinje cells, a guy named Purkinje first described it, that these Purkinje cells in any one bit of cortex, that they're lined up, where they are very evenly spaced apart, and their dendrites, those fingers, are only stretching out in 2 dimensions, and they're all parallel to each other. But how did that happen? Well, we're going to tell you, right? So in the cerebellum, then, we have the ventricular zone. The intermediate zone is where eventually all the axons are going to be, and they're going to be myelinated. So they're going to make the white matter. The intermediate zone turns into white matter. And now we have the outer zones that are going to make all the gray matter. And in cerebellum, we'll have an internal layer of granular neurons. Why are they called granular neurons? Because they're tiny and sort of like little grains. And then there are the massive Purkinje cells with their dendrites sticking up. And then there'll be another layer of granule cells at the pial surface with the marginal zone in between. Okay? And eventually we're going to talk about how this arrangement comes about as cells are shitting around and sorting each other out. In cortex, the terminology is a little bit different. So in cortex, again, we have the ventricular zone, but then there's something known as the subventricular zone. And in the cortex, pretty early on, this division happens. And so now all the division is actually taking place in the subventricular zone. So when I said there's division going on in your ventricular zone right now, I was telling you the truth slowly. Specifically, in your ventricular zone, it's the innermost portion of the ventricular zone that we call the subventricular zone. That's where the division's going on. The ventricular part of the ventricular zone that's right next to the ventricle wall, they're not dividing so much, and they're just there to protect the brain from stuff that's in the cerebrospinal fluid. It's incredibly confusing that the field calls this bunch of cells above the ventricular zone the subventricular zone, but you're stuck with it. I didn't do it. Nobody asked me. Okay? So in cortex, all cortex, there'll be a ventricular zone in adults and a subventricular zone. There will be the intermediate zone where the axons of all these neurons that shinnied up have gotten left behind, and which will eventually make white matter. And in the cerebral cortex, there aren't Bergmann glia, but there are another kind of glia that, like the Bergmann glia, are attached to both the peel and ventricular surface. And the neurons, when they kick out of mitosis in the subventricular zone, they climb up these cells to get to where they want to go. The glia are called radial glia because they look like radii, spokes in a wheel. I'll show you another example later. So the cells are climbing up the radial glia, and now they're going to make what we think of as a cortex, and during development, it's called the cortical plate. It hasn't yet divided into 6 layers, but we're going to see the 6 layers soon. And in the meantime, the marginal zone gets left out here, and eventually there'll be another layer that consists only of synapses and axons and dendrites. And so that's known as the molecular zone, because when the early histologists looked there, you never saw any cells out there. These two cells together are going to make layer 1 of cortex. And layers 2, 3, 4, 5, 6 are all going to be, are eventually going to coalesce here in the cortical plate. Okay? Let's see how this works. So, and how we know about it. It turns out early in development of radial glia, actually early in development of glia, there's a particular protein that they make that is called glial fibrillary acidic protein. And only glia make it. Neurons don't make it. And that is so convenient because pretty early on, say, 1970s or so, someone made an antibody that recognized glial fibrillary acidic protein, GFAP. And that meant they could use this antibody to identify glial cells in developing nervous systems. And that's when they realized there were these radial glia, right? They were spanning wide distances. And at first, the radial glia are progenitor cells. They're going to give rise to the entire nervous system in this area. So they're doing that thing, that shuttling of the nuclei, and they divide down here. And when they divide, they make 2 more radial glia. And then the nuclei go up before they come back down for the M phase, right? We are making the M phase, the mitotic phase of division. So far, so good. And then eventually asymmetric divisions start happening. And eventually the divisions keep going on here, and the asymmetrical partners start shinning up the radial glia to get to the peel surface. At this point, layer 1 is already there and is present. But what happens is these neurons, they shinny up and they stop just short of layer 1. And later, neurons are going to come and go past those cells and also stop at layer 1. More about that later. During development, then, there's a particular, this note is to point out that when division takes place, neurons don't divide. So there's no such thing as neurons dividing in normal cells. But there are cells that divide, and right away one of the cells makes a neuron, so we call that neurogenesis. And if we're talking about division that results in glia, we call it gliogenesis. So as in this case, in this diagram, what we're trying to show is that at first there's only radial glia, and they're only dividing symmetrically, making more radial glia. And then eventually asymmetric divisions start happening, and the first cells that get made are the neurons. And as things continue, now the dividing starts moving from the radial glia to the subventricular zone, where they're not radial glia, they're just cells, neuroblasts, and they're dividing like mad. And the ventricular zone is still there, but that's not where the divisions are going on. Here's the intermediate zone that's eventually going to make the white matter. And here are the cortical layers. And what we're trying to show you here is that the lower cortical layers get formed first, and then the upper cortical layers, with the exception of the marginal zone, layer 1, which stays there all the time. Are you exhausted yet? Yeah, okay, let's do a Kahoot then. And you can ask questions while it's coming up. Realize that there isn't more variety in the treats.
This.
Okay,
I'm a certain.
so we're going to take time to do a little bit of methodology here to keep track of birth dating of cells, specifically birth dating of neurons. You remember that DNA is made up of 4 nucleotides, right? And one of them is thymidine. But RNA doesn't use thymidine, right? Instead, it uses uracil, right? So that means that thymidine is mostly found in DNA. And so back in the prehistoric days of the 60s and 70s and 80s, you could get molecules of thymidine and radioactively label them. Make one of the atoms radioactive, like make one of the hydrogen atoms tritium, or make one of the carbon atoms, carbon-14, something like that. Everyone got that? And it has a half-life, right? I mean, eventually the radioactive particle will give up its the radioactive molecule will give up its particle and release, a particle. So if you inject an animal that's developing with radioactively labeled thymidine, how much of it will go to RNA? Right, none. Instead, it will get used for DNA. And in an embryonic individual, it's sucking up thymidine all the time, because with every division, you got to have a lot of thymidine molecules, right? And so you can inject radioactively labeled thymidine into, say, a pregnant rat when you know her offspring are in the 12th day of gestation. And so in those fetuses, any cells that are dividing are going to suck up a bunch of that radioactively labeled thymidine. And once they stop dividing, that thymidine is there forever, for the rest of the animal's life. So now you have a marker for, this cell, and you can look much later into adulthood, this cell must have been dividing or at its last division or near to last division back there on day 12 of gestation when I injected the stuff. Everyone get the idea? But you might be saying, well, wait a minute. What if it's early enough in development, that cell's going to divide a whole bunch and produce lots and lots of offspring, right? But remember, if there's a lot of divisions that happen thereafter, the thymidine is going to get diluted so that those 1,000 offspring, you won't be able to find it anymore. So the only cells that will be labeled that were either undergoing their last cell division, specifically they're at the S phase, synthesis phase, where the duplicate DNA molecules are getting made. They were either at their last division or near last division, back on day 12 of gestation. You get the idea? And by doing this, people discovered things like the first post-mitotic neurons in the spinal cord are motorons. Why? Because you have to inject the thymidine by the 12th or 13th day of gestation. If you inject it any later than that, motorons are never labeled. And if you inject it too much earlier than that, they're not showing a label anymore because the signal got diluted. And that's how we know that neurons in the spinal cord, motorons are the first neurons that arise. There's something comforting, by the way, that if you inject, say, on the 15th day of gestation, you never label any motorons. What's comforting about that is you might be thinking, well, I've heard about stuff like DNA repair. What if cells take up the thymidine because they're doing some repair in their DNA and they just need a little bit of thymidine? And indeed, some of that must happen. But there can't be very much of it happening because when you inject thymidine in adult individuals, no neurons ever take up the label. So it really is a marker for mitosis, not for DNA repair. In other cases, in other animals, say like leeches, or, you can't do this in C. elegans because their cells are too small. But in leeches, for example, they have really big cells that are dividing, and so you might inject a marker, like a fluorescent marker. And if a lot more divisions happen in the cell you injected, the marker will get diluted. But if only a few divisions happen, then you can keep track of that fluorescent marker and see what its offspring became. How's that? So if you are using radioactively labeled thymidine, you inject on the 12th day of gestation, you wait till the fetuses grow up and you kill them as adults, and you look at their spinal cord and you see, lo and behold, these motorons have thymidine inside them. And to know that, you have to slice up the spinal cord and put the slices next to photographic film. And then as the tritium gives up its particle, it will expose the film and leave you a nice little silver grain over the nuclei, the motorons, like that. Total pain in the ass to do. That's the technical term. For a lot of reasons. Anyone know the half-life of tritium? It's like 10 years. So, for half of the tritium molecules to give up the particles, you'd have to wait a long time. So you got to inject a lot of radioactively labeled thymidine, not tritium. And plus, you've got to slice this tissue and then get it next to photographic film. And as soon as it's next to photographic film, you have to keep it shielded from light. And you got to wait, say, 3 months, 4 months, before you take the slides out, photographically develop them, and see if your experiment was a total bust or not. Really annoying. And sometimes it is, right? Total bust, I mean. And now you know, oh, you did something wrong earlier. This business of using thymidine to tell when during development a cell came to its last divisions is known as birth dating. So this cell divided and it never divided again, and so this is its birthday. This is when it kicked out of mitosis and took on a life as an adult or as an individual neuron. And the whole thing, the whole business got revolutionized when people realized that there was a molecule that did not occur in nature that was enough like thymidine that cells in S phase would take it up to make their DNA, but was different enough from thymidine that you could make an antibody that would recognize it. And so now, instead of injecting radioactively labeled thymidine and waiting months to develop it, you inject this stuff, bromodioxyuridine. You can tell from the name that's kind of like uridine, but it doesn't get taken up in RNA, only gets taken up in DNA. So it is a DNA-specific marker. And so you inject mom in the 12th day of gestation, you wait for the pups to grow up in adults, you kill the animals, and now instead of looking for photographic plates, you simply use an antibody to stain the tissue and see where the BrdU was. And wherever the BrdU is, that's a cell that was in its final or near final mitosis back when you injected the stuff. So nobody uses tritiated-thymidine and autoradiography to keep track of birth dating anymore. You're looking at one of the few people left in the world that knows how to do it. And the reason nobody else knows how to do it is because nobody would ever want to do it. It's a dumb idea. And you see the advantage, right? You still got to wait for the animal to grow up. But once you kill the animal, you get to see the results in just a couple of days without having to work with a sharp instrument, sharp knife in a dark room. Okay. So when we get back, we'll see that once people use these birth dating methods, they realize there was a very specific pattern of organization in the development of the 6 layers of the cortex, the cerebral cortex. Okay? See you then.
I just wanted the correct.
The thila. Yeah. I should have done that because
So I mean, there are a lot of kids in this class. I don't know how you.
well, no, I mean it needs to end with an A because it's a girl's name, right?
Yeah, I